So, hello everyone, good evening. So, my name is Radharani Benvenutti. I am postdoctoral researcher here in RCSI and PhD in neuroscience. And today I will talk about the research that I have been developing with the support of the Epilepsy Ireland and the IRC and the FutureNeuro, the circadian biomarkers in preclinical and clinical Dravet syndrome. So in this presentation, I will give you a brief overview about the circadian rhythms and their relation to Dravet syndrome. I will show you the project aims and the methods and the expected results for this project. But first of all, what is the circadian rhythms? So, as you know, the environment around us is always changing, and daily we are exposed to darkness and light and cold and hot temperatures. We feel hungry, we feel sleepy, and our body needs to keep responding to these changes to be adapted to the different daily situations and stimulus. But how does our body know what time is it? So we have a biological system that will be always working on that. This is the circadian rhythm. And this is a complex system responsible for coordinating all the daily changes in physiology and behavior across 24 hours. In the system, we have a master clock in the brain. This is the suprachiasmatic nucleus that is able to catch all the signals from the environment, external and internal, and will prepare our body to be adapted to all these changes. So if the circadian rhythm is working well, our body is able to anticipate daily changes in the environment, including light exposure, food availability, and temperature, to have the optimal fitness and adaptation of our body. But in addition to this master clock in the brain, we know that each cell in our body, in our tissues, will have its own clock. So exactly like a conductor leading an orchestra, the suprachiasmatic nucleus in the brain will synchronize all the other clocks in our cells. Sometimes the circadian rhythm can be dysregulated, and this is what we have been observing in several diseases, including epilepsy. So, in spite of the fact that we thought that seizures were random, actually they are not. And around 80% of people with epilepsy present a circadian rhythm mystery in their seizures. That means that some people are more likely to have seizures only during the day, while others are more likely to present only during the night. And this is very relevant for Dravet syndrome, a rare type of difficult-to-treat epilepsy. It's known that people with Dravet syndrome largely present sleep disturbances and nocturnal seizures, which seems to increase the risk of SUDEP, sudden unexpected death in epilepsy. In spite of this evidence, we still don't know how the clocks are ticking in Dravet syndrome. Therefore, in my project, I am investigating whether the regulation of these clocks could be linked to the development of seizures and to the increased risk of SUDEP. So more specifically, I am addressing this question in two parts. So in the first part, I am using a mouse model that mimics Dravet syndrome to map all the circadian rhythm in brain and blood. So I am using this mouse that presents the same mutation in the gene SCN1A that most of people with Dravet syndrome presents. And so I will understand. I also will identify which genes and proteins change their expression in a circadian way. And completing this first part, I expect to have identified a number of possible targets that could potentially be relevant in humans. Finally, this will be validated in a second part of this project with the support of international collaborators. So here, I will recruit people across multiple clinical sites in Europe, and I will use a number of tools, such as the application of specific questionnaires and assessments like actigraphy and EEG, the electroencephalography, and also blood samples to profile the circadian rhythms regulation in people with Dravet syndrome. So altogether, this data may be helpful to understand mechanisms that may be involved in the disease progression and SADAP. Overall, in spite of shining a light about the role of the circadian rhythm in Dravet syndrome, I expect that my findings can support future studies to predict SADAP risk or to develop new treatment strategies. So I would like to thank you all for your attention today, and also to all the collaborators in this project, mainly the FutureNeuro Center, Epilepsy Ireland, and the Irish Research Council, and mainly to my supervisor, Christina Rush. Thank you so much.